Certain phagocytic cells of the immune system, called antigen-presenting cells, seek out foreign cells and engulf them. In addition, despite the best efforts of the immune system, cells can become infected. When this happens, the immune system needs a way to see inside the cell in order to know that it is infected or has found invaders. Mammalian cells use antigen processing and presentation to accomplish that task. Some of the protein molecules synthesized within most nucleated cells are digested into smaller peptides, about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins, which are products of a group of genes called the major histocompatibility complex. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol. Vesicles containing the MHC peptide complexes travel through the cytosol to the membrane. The vesicles fuse with the membrane, delivering the complexes to the exterior of the cell. MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow cells of the immune system to detect proteins inside the cell without entering the cell. Cytotoxic T cells are the warriors of the immune system and locate and destroy infected or cancerous cells in the body. They are activated by infected cells and the cytokines produced by helper T cells. Active cytotoxic T cells proliferate, producing additional active cytotoxic T cells as well as memory T cells. Active cytotoxic T cells destroy their targeted cells through cytotoxic pathways, such as the perforin granzyme pathway. Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants. Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present antigenic determinants on the surface of the cell. The association of the antigenic determinants with specific MHC proteins signals immune cells that these antigenic determinants came from digested invaders. Certain immune system cells, such as macrophages and neutrophils, utilize phagocytosis to ingest and destroy invading microorganisms. These phagocytes attack and destroy invaders using chemotaxis, adherence, ingestion, digestion, and elimination. Chemotactic chemicals attract the phagocytes to the site of infection, and opsonins aid in adherence of the phagocytes to the surfaces of the microorganisms. Helper T cells are the generals of the immune system and are responsible for directing the activity of the immune system. They are activated by antigen-presenting cells. Active helper T cells proliferate and differentiate into memory T cells, as well as two populations of helper T cells, Th1 and Th2. Th1 cells primarily activate cells related to cell-mediated immunity, while Th2 cells primarily activate cells related to humoral immunity.